This is really a privilege on my part to share some of my views and experiences working with SARS-CoV-2, particularly highlighting the complexities in the viral genome and the prospect of its control. Before I start, I would like to express my deepest sense of gratitude to Dr. Devina Nair and also the principal of DKD College for inviting me and giving me this opportunity to share my views with the participants. I think by this time, all of us are aware of the menace of COVID-19 and the SARS-CoV-2. I'll try to highlight some of the important aspects on the complexities of the profile of the virus, particularly in its genomic sequence, and try to correlate the features of the virus with the prospect of its control. As we all know, that the coronavirus, which is now causing the pandemic all over the globe, was first detected in China on the last day of 2019. And thereafter, it has spread now to more than 213 countries of the world, killing more than 7.47 lakh people and affecting 21 million people all worldwide. And the numbers are growing daily. As you all might be knowing by this time, that there are a lot of things about the virus which are yet to be clear, because this is totally a novel virus. Although coronavirus family was known since long before. Coronaviruses are a large family belonging to the family Coronaviridae, and many of the components of this family are regularly causing diseases in human and animals. To name a few of such diseases, the common cough and cold, which is encountered in most parts of the world seasonally. The novel coronavirus is a relative of two more hitherto well-known coronaviruses. There is severe acute respiratory syndrome virus, or SARS coronavirus, and Middle East respiratory syndrome virus, MERS virus. Now, if we go into the general feature of the coronavirus structure, we find that in every coronavirus, there is a core genome admixed with the protein capsid or the covering, forming the nucleocapsid, and which contains this important protein called nucleocapsid protein, designated as N protein. And it is an envelope virus, therefore it is also covered by an envelope. An envelope has got also certain projections or whiskers from its surface, which are also called spikes. So this constitution of the virus gives the typical structure of the virus like the crown of a king, and that is why the name has been given as coronaviruses, because corona means a crown. Now, this virus is one of the largest RNA viruses, and the total genome size is around 27 to 32 kb, and it has got 4 prominent proteins in it. There is nucleocapsid protein, which is referred to as N protein, membrane protein or M protein, envelope protein or E protein, and spike protein or S protein. Now, like other coronaviruses, this virus is also spherical, and the spikes play a very vital role in attachment of the virus to the surfaces of the mucosa of the organs affected by the virus. This particular virus with its spikes can easily bind to certain receptors on the human cell named as angiotensin converting enzyme 2 or ACE2. And interestingly, the efficacy of this virus to bind to ACE2 is around 10 to 20 times more in the novel coronavirus causing COVID-19 in comparison to earlier known coronaviruses causing temporary pandemics like SARS or MERS viruses. That is why it is causing so much of severity in infection all throughout the globe. And as I already say that now the COVID-19 is occurring in more than 213 countries and territories of the world. And the attack rate or the transmissibility of the virus is much more than the commonly known viruses causing similar kind of widespread infection like your influenza virus, or for that matter other coronaviruses causing similar kind of pandemic earlier, as I already referred, SARS and MERS. So if you consider the transmissibility or the ability of the virus to transmit from one individual to others, it can be represented in case of SARS-CoV-2 or the new coronavirus as transmissibility between 1.5 to 3.5. That means you can say that from one single individual, it can readily transmit to around 2 to 4 individuals on an average. But while you compare this transmissibility to other viruses like common flu, the common flu is having the RO value or transmissibility value of 1.3, and SARS was having the transmissibility value of 2. On the other hand, the case fatality rate or the mortality pattern or the death rate induced by this virus is much lower than that of SARS and MERS, because MERS was more fatal, causing around 34% mortality among the patients, whereas the SARS was causing 10% mortality among the infected individuals. While globally, the trend is the COVID-19 is causing around 2 to 3% mortality. And in India, it is even lesser than many other countries. And in Assam, so far, mortality has been found to be very low, only around 0.24%. This is quite a significant finding in the state of Assam. And in India also, it has come down below 2% yesterday. Now, if you consider the virus property, another unique property is that after getting infected, an individual may develop symptoms only after 2 to 14 days of infection. So this is referred to as incubation period. That means after infection for up to 14 days, the patients may not show any symptom. But most of the time, there's a question or query in the minds of people whether during the incubation period, the virus can be spread by the infected individuals. The answer is yes. Means even 3 to 5 days before showing symptoms, an individual infected with SARS-CoV-2 virus can transmit the disease to the susceptible individuals by direct contact. So in some of the cases, some unusual pattern of transmissibility was also seen, wherein there has been some reports of development of symptoms even after 27 to 28 days of infection or exposure to infection. So that means we can say that the incubation period may also vary greatly depending upon the dose of the pathogen that is introduced into the susceptible individuals. As all might be knowing by this time that the mode of transmission of the virus is basically through direct contact through the droplets. That means when an infected individual coughs, sneezes, or even talks, it can be spread through the droplets coming out through the mouth and nose to the neighboring areas. And if it can somehow reach the nose, eyes, or mouth of susceptible individuals around, then they can get infected. Now, why it is called droplet? Actually, small molecules, small particles of water, less than 5 micrometer or 5 micron in size, are usually referred to as droplet nuclei. If the virus size is smaller, then it can spread very easily with a small droplets of water molecules or water vapor less than 5 micrometer in size. And in that case, it can spread through air very easily. Airborne transmission is possible very easily. But in case of SARS-CoV-2 virus, it is not usually regarded as very commonly spreading through airborne transmission because it is a larger size RNA virus, mostly is transmitted through larger size droplets, means more than 5 micrometer size. If you see this picture, it will be clear to you. You see, these are the large droplets of water molecules, which contains the viruses of COVID-19, SARS-CoV-2, which can only get spread to a neighboring place. So maximum is around, say, 6 feet. That is why a social distancing of 6 feet or more, or at least 2 meters, can prevent you to a great extent from exposure to infection coming from the droplets in the form of droplets from the infected individuals through their coughs, sneezes, or coughs. But in airborne transmission, when they can transmit through small droplets, nuclei, less than 5 micrometer in size, then they can spread to longer places or distant places, and they can be carried by wind also. Interestingly, although at the beginning, the SARS-CoV-2 was not thought to be spread by airborne transmission, but it has been already recognized by WHO recently, around 15 days back, that outbreaks can also occur due to spread of the virus through airborne transmission if people are overcrowded in a small space indoors and there are some infected individuals. Then it can spread through small droplet nuclei by airborne transmission. And it has been also postulated that the small cabinets full of people, individuals, and if it is also having air conditioner, the spread may be always more than open, if there are open spaces or if the windows of the rooms are not closed. Now, if you see the variation in terms of its pathogenicity in relation to age and sex, then you will see that already globally a trend has been recognized wherein it was found that out of 100 individuals affected, mostly around 50% of the individuals are within the range of 75 years and old. I'm sorry, not affected, dead. Most of the people dying of the COVID-19 infection are within the range of 75 years and more. So around 50% of the individuals who have died, they are of this particular age group. And in younger age group, lower the age group, the mortality is lesser. And similarly, the number of infections are also lesser. So in one particular study that was done in New York City, it was found that there was no mortality or fatality in people less than 9 years old, but with the increase in age group, the fatality or the death rate was higher, having the maximum fatality rate in the 80+ years old individuals. So that is why the WHO as well as the ICMR has given a guidelines that those individuals who are more than 65 years of age should remain indoor and they should not expose themselves to the crowded population or infected individuals. So for safety, because the infection rate and the death rate is also higher in aged individuals. If you see the pattern of distribution according to sex, you will see that globally males are more commonly affected and death rate is also higher in case of males. But the clear, so to say, region for this variation in the age distribution pattern is not yet understood. But it is assumed that this is only because of probably the more exposure of male individuals because of their professional or business related activities outside the home. They are more affected and their mortalities are also more. Now the question is, why does some of the individuals suffer in a particular environment more than others. In a particular community or in a particular small place, even within a family, there may be variation in terms of pathogenicity. Some individuals in the group or the family may suffer from the infection, but others may not be affected, even if they are living in the same environment. Why this is so? This is usually affected by many different factors, including social and demographic factors, including sex, race, ethnicity, income, and access to quality healthcare, and many other related confounding factors. But some other important factors have been identified scientifically, which may also play a role in transmission of the disease and occurrence only in particular group of individuals. Say, for example, it has been found that there are more than 22 different proteins which consistently appear in large quantities in severely infected patients, which are not commonly found in large, so to say, extent in the unaffected individuals or those who are suffering from mild infections. So these 22 proteins may be quite relevant in terms of its relation to the pathogenicity of the virus, but it is yet to be ascertained what are the importance of these proteins in the pathogenicity or the virulence pattern of the virus. On the other hand, there has been some reports of lesser occurrence of the disease in individuals with O blood group and more occurrence of the disease in type A blood group individuals. So in a particular research work conducted in US, it was found that around 50% of the individuals may be having higher risk of needing oxygen and ventilator if they suffer from COVID-19, if they are having the blood group A antigens, if they are of type A blood group. Similarly, the people having type O blood group, the universal donor blood group, are usually having 9 to 18% lesser likelihood of positivity of COVID-19. But of course, we cannot be complacent in this. Those who are having O blood group, they should not think that they are resistant to the infection, because you see the mere percentage of 9 to 18% less likely to suffer. So that means you are also likely to suffer by 91% or more. So that's why it is not a matter to become complacent that if you are having O blood group, you are not having the likelihood of getting transmitted. Then there are a lot of research work going on at the present time to see the variation in the whole genome sequences of the viruses. It has been already identified that major strain variations are occurring in the viruses in different parts of the world. The whole genome sequencing of thousands of different strains of viruses from different countries of the world have been already conducted. And based on the result of such studies, it has been found that the virus has already shown a lot of mutations, and divergent group of genotypic population have been already identified. The severity of the illness of an individual will therefore depend upon the particular virus type or the genotype of the virus by which it is affected. But a clear, so to say, relationship could not be established so far regarding a specific mutation occurring in the virus in relation to increase or decrease of virulence. But even then, it has been already established that the virus is undergoing mutation, and there has been a lot of genotypes occurring in the population of the viruses in different countries of the world. One more important point to emphasize is that, as I already said, that the severity of the infection of an individual will not only depend upon the type of the virus involved, but also will depend upon the amount of the virus initially transmitted, or amount of the virus which has been inhaled or getting exposed by an susceptible individual. Normally, it has been found that the deaths in the severely affected patients mostly occurs due to occurrence of an overactive immune response, what is referred to as a cytokine storm at the terminal stage of the infection, causing death of the individuals. And this has also, so to say, given rise to the specific, so to say, treatment regime in severely affected patients who are in ICU or who are in oxygen support, to be treated with steroid, because steroid can be used to get rid of such kind of overreactive immune response, preventing the cytokine storm, which may be helpful to avoid death. So nowadays, steroids are usually applied in terminally ill patients to prevent death in severely ill patients. Now, mutations, as I said, have been already reported in the virus in different parts of the globe. An analysis of more than 15,000 SARS-CoV-2 genomes could find that mutations to the virus do not increase its transmissibility. So far as transmissibility pattern is concerned, it could not be clearly established by looking into the genomic variations. When a comparison of the SARS-CoV-2 was made with other coronaviruses, including SARS-CoV virus and MERS-CoV virus, it was found that in comparison to other coronaviruses known, the similarity to SARS-CoV-2 is more to the SARS-CoV virus, which was occurring as a pandemic in 2003, and MERS-CoV virus, which occurred as another pandemic in 2009. Now, genomic diversity could also establish that the SARS-CoV-2 is more related to the bat coronavirus and coronavirus of Malayan pangolin than other viruses, coronaviruses of different species of animals, including human. So this has been clearly established beyond doubt that the ancestor of the SARS-CoV-2 might have been the bat coronavirus of the same group of coronaviruses of 4 genera, 4 different types, alpha, beta, gamma, and delta. And SARS-CoV-2 and the SARS-CoV virus both belong to the beta type, and the bat coronavirus belongs to the alpha type. And there has been high level of similarity between the beta and alpha type of coronavirus. That means the SARS-CoV-2 virus is showing highest level of similarity to bat coronavirus and then Malayan pangolin virus. Then the multiple clusters or clades have been detected among the coronavirus population circulating in different parts of the country, as I said, and it is showing 99 to 100% similarity to other coronaviruses. Particularly 93% homology was found with bat virus, and 84% homology was found in case of Malayan pangolins. Now you see, as I already described, that beta coronaviruses have already undergone some mutations causing changes in their genotype, and there are 4 lineages which have been already established: lineage A, lineage B, C, and D. And SARS-CoV virus and the SARS-CoV-2 virus, means the virus of COVID-19, belongs to the lineage B. That means it is most commonly related to the SARS-CoV virus, which occurred as a pandemic in 2003, and it is also little closely related to MERS-CoV-2 up to the extent of 88% homology have been found. And if we now examine the whole genome sequence of thousands of coronaviruses, we also see the genomic variation among them occurring due to mutation. This is one study made very recently in June 2020, which was published in PNAS, done by Forster and co-workers, based on the analysis of the whole genome sequence of 160 SARS-CoV-2 viruses, where it was found that the original SARS-CoV-2 virus, which occurred in China and some other countries of the world, which were having close relationship with the bat coronavirus, and they clustered in a small group of cluster, which was known as a cluster A. This is cluster A. Then they deviated from the cluster A by mutation, and another cluster has been formed, which is known as cluster B. Then from cluster B again, there are a lot of mutations going on in the virus, forming another new cluster, which is designated as cluster C. Now, definitely, they have identified 3 distinct clusters among the whole genome sequence of the coronaviruses causing COVID-19. And it has been found that in India, most of the viruses are belonging to this cluster B. And in some other countries of Asia, some component of the cluster A, B, and C can be found, although the original cluster was only of the cluster A. Then if you look into another report of similar kind of whole genome sequence study, which was done by Mandal and co-workers in 2020 in India, based on 449 genome sequences of the SARS-CoV-2 virus, that means downloaded from NCBI website, which represent different strains of the virus in circulation in different parts of the globe, or different continents of the globe. It was found that the SARS-CoV-2 virus, which is circulating in India, are mostly confined to 2 clusters, that is cluster I and cluster Z, clade I and clade Z. They are closely related, and they are similar to the viruses occurring in only few other countries of South Asia or East Asia. But most of the other countries in the Far East or in the Western countries, they do suffer from other viruses of other clusters, and they do not have, that means, close similarity to the circulating viruses in India. So a lot of mutations have already occurred in the genomic sequence of the virus, and we can identify the viruses circulating in a particular population by identification of the specific mutation occurring in the genes or genome of the virus. And based on that, phylogenetic tree can be built, and this has clearly shown that the Indian strains of the SARS-CoV-2 virus belong to the clade I and Z most commonly. Another report which has very recently been published by Banu and co-workers in June 2020 from a group of scientists from CCMB Hyderabad, Center for Cellular and Molecular Biology Hyderabad. They found an interesting finding. So in their finding, they found that the strains of the virus, which are in circulation in states like Maharashtra, Tamil Nadu, and Delhi, they can be grouped in a new clade of the virus known as clade A3I. It is belonging to the cluster I as a whole. I and J, as I said, belongs to the cluster I, but it has a small clade of the virus, a small group of the virus, which is distributed in the clade A3I. But in other parts of the country, in other states of the country, mostly it is of A2A. And of course, few reports of A3, B, and B4 have also been reported. So this peculiar distribution of viruses belonging to clade A3I is significant because it has been found that the severity of infection in Maharashtra, Tamil Nadu, and Delhi is higher than other parts of the country. And in some areas, the death rate is also higher in Maharashtra and Tamil Nadu, going beyond 7%, as that of some other countries like Italy, Spain, or in US. But in India as a whole, the death rate is usually lesser than 2%. So now it is assumed, but of course it is not clearly understood how this variation may be related to the virulence of the pathogen or the unique kind of mortality or severity of the disease occurring. But even then, it is assumed that the severity of the disease or the increased mortality in the population of Maharashtra, Tamil Nadu, and Delhi might be somehow related to the specific genotype of the virus or the genetic characteristic of the virus in circulation in those states. So this is the phylogenetic tree that has been established by the report, wherein, as you see, the strains which have been isolated from those three states mostly belonging to this clade A3I, whereas the strains from other parts of the country belong to A2A. Now, coming to the control of the disease, control is possible by treatment of the infected individuals and by means of protecting the individuals by way of vaccination. Now, while we discuss about treatment, as we all know that it is a viral disease, and therefore there is no specific treatment as that of any other viral disease. But of course, in terminally ill patients, if required, some antiviral drugs can also be tried to spare the life of the individuals. There has been a lot of research going on all throughout the globe to find out some novel treatment regime for the COVID-19, because it is a new virus, no known treatment was available. 35 drugs are in the race to treat COVID-19, and a lot of reports are coming up about efficacy of different treatment. But usually, as per the ICMR guideline, in patients suffering in India, while they show symptoms, when the symptoms are milder or very minimum, then they do not prescribe any specific treatment, only symptomatic treatment, particularly paracetamol or some antibiotic. But in severely ill patients or moderately ill patients with oxygen saturation below 95%, some treatment can be prescribed. There are a lot of investigative therapies prescribed by ICMR now. Out of those, investigative therapy of remdesivir has been already started in all states of the country, although it is under complete supervision of expert medicos, and it is already defined as an investigative therapy because it is not an established therapy as of yet. So it is an experimental therapy. Only in severely ill patients they are tried with certain guidelines to be followed. And another treatment is being followed is your plasma therapy. The convalescent plasma is being used because use of hyperimmune serum or plasma of recovered patients were known since last several centuries, and in many diseases the plasma or serum of individuals who have already recovered from disease was used to treat cases. This has been also found to be helpful in saving the life of severely ill patients in case of COVID-19. And in Assam also, the government has initiated the use of convalescent plasma for treatment of severely ill patients in the ICU. Many other drugs are in the, so to say, trial, like favipiravir, umifenovir, and many others. And at the beginning, many experiments were also showing success of use of hydroxychloroquine, an anti-malarial drug, in the treatment of COVID-19. But later on, it was found that this is not a very effective drug. Rather, it causes some severe side effects in terminally ill patients, and it may also increase the death rate of terminally ill patients. That is why, in many countries of the world, the use of hydroxychloroquine has been discontinued. As I already said, dexamethasone or other steroid therapy can also be applied in the patients, and particularly those who are in oxygen support and in ventilation, to save their life, because as I said, in terminally ill patients, a cytokine storm can be prevented by using steroid. And of course, aspirin has also been used in some patients. Particularly, it has been advocated by scientists and doctors in Italy to use aspirin in the severely ill patients to get rid of the clotting of blood in the terminally ill patients, causing death. So this can be prevented by using aspirin, and it was very highly advocated by the doctors in Italy. Then coming about vaccines, a lot of different vaccines are under trial in different stages of trial. 8 different types of vaccines are under trial. They may be inactivated vaccines, weekend vaccines, or nucleic acid vaccines containing DNA or cDNA, complementary DNA sequences or RNA sequences of the virus. There may be viral vector vaccines. Either it can be a replicating vector vaccine or non-replicating type of vaccine, protein subunit vaccines, and virus-like particle vaccines. Different types of vaccines are investigated. Most of the vaccines which are in different levels of trial are either viral vector vaccines or protein-based vaccines. There are several vaccines under trial. As has been reported in a paper recently, 153 different vaccines are in trial in different countries of the world. But out of them, these 6 or 7 vaccines are already in the race, and they are the front runners. And they have succeeded in showing promise in the first phase of the clinical trial. As you know, after the preclinical trial in the animals, the clinical trial in human being is conducted usually in 3 phases: phase 1, phase 2, and phase 3. Most of these vaccines, which are shown here as front runners, have either completed the phase 1 trial or phase 2 trial, or phase 1 and phase 2 trial are now on. But no vaccine has now completed the phase 3 trial. We should mind it. The first vaccine, which has been registered very recently, only 3 days before, on 11th August, as you all might be knowing, was in Russia, which is known as Sputnik 5. This vaccine has been approved by Russian government, but of course, this has already completed the phase 1 and phase 2 trial, but yet to complete the phase 3 trial. Although it was showing a great promise, but there has been a lot of controversies regarding this vaccine, and many people say that it may have very severe side effects. And as has been reported by a paper very recently, that as many as 44 different side effects could be detected in phase 1 and phase 2 trials. So it is really doubtful whether this will capture the market all throughout the globe. Another very promising vaccine is now in the phase 2 trial in advanced stage, that is the Oxford vaccine, which is full name is called CH80OX1 vaccine. And this has been done by Oxford University of Oxford in collaboration with another private company, AstraZeneca. And this has already completed phase 1 and phase 2 trial. And interestingly, they have already become prepared with 2 million doses of the vaccine already available. Once the third phase of clinical trial will be completed, these 2 million doses will be immediately applied in European countries, particularly England. And of course, they have also requested the Serum Institute of India to produce vaccine, and they have already come up with their so to say, support to produce this vaccine in large numbers once it becomes successful. Similarly, another American vaccine, which is called Moderna COVID-19 vaccine. This is a messenger RNA-based vaccine called mRNA 1273, based on the spike protein of the virus. This has also been found to be highly promising. Already they have completed the phase 1 trial. The phase 2 trial is in process now. And they have already given order to the Serum Institute of India to produce 10 million doses of this vaccine. And Serum Institute of India is known as the globally front runner in vaccine production. Most of the vaccines which are available in all countries of the world comes from Serum Institute of India, and they have already taken the contract of 10 million doses of the vaccine. Similarly, Pfizer BioNTech, this is another vaccine which is also in phase 2 trial. This is also a promising vaccine. Another vaccine is recombinant VSV Delta Z spike vaccine. It is from Israel. This Pfizer BioNTech is a combined joint effort of France and Germany, and this is an Israel vaccine. Similarly, India has also initiated indigenously producing a vaccine with the help of a company, Borah Biotech, in collaboration with National Institute of Virology and ICMR. And it has already started phase 1 trial, and it is expected that in next 2 to 3 months' time, the Covaxin, the indigenous Indian vaccine will also come to the market. It is expected that it will be available to the Indians at a very low cost, and already it has been declared that it will be costing even less than 250 rupees per dose. WHO, World Health Organization, is highly hopeful of getting the control of coronavirus infection through vaccination by the end of 2020. And as I already said, billion doses of vaccines are already in the process of manufacturing by different agencies and companies. And at the beginning, of course, when the vaccine will be coming to the market, probably the individuals at high risk will be getting the advantage of getting first vaccinated because they are the front runners in the fight against COVID-19. The medicos, the health workers, then the security personnel will be getting the first preference of getting the vaccination. And of course, thereafter, it will be available for the general public. Another question in relation to protection of individuals is herd immunity. Now, many a quarter usually many times say that the real way of getting protection in India or in several other third world countries where the population is very high is only due to development of herd immunity. Because even if the vaccine is made available within the next 3 to 4 months, it may not be available for the common man. And the government may not be able to afford universal vaccination for all the people in the countries like India and other third world countries. And probably it will not be possible to immunize all individuals within a period of 1 or 2 years. And even there is doubt whether the vaccines will be really protective to the extent of 100%. And there may be strain variation, as I already said, and all vaccines may not be equally effective in protecting individuals against all different genotypes of the virus. That may also be found later on after introduction of the vaccine, that some of the vaccinated individuals may also suffer from infection. So nobody can be certain at this point of time whether one or the other type of vaccine will be 100% successful in giving protection to the vaccinated individuals. Therefore, many scientists also believe that development of herd immunity is the only answer to giving protection to the millions and billions of individuals in third world countries. To be precise, what is herd immunity? Herd immunity means is a level of protection induced when a large proportion of a community in a population becomes immune to the disease. This immunity may be achieved either through natural infection or through vaccination. Now, if it is acquired through vaccination, it's well and good because vaccination is the most important, so to say, way of inducing herd immunity. But at this point of time, as I said, when we don't have vaccine, only way to get herd immunity in a population or community is to get infected in large numbers. So some scientists have already said that maybe in countries like India, Bangladesh, or other populated countries like China, maybe unknowingly, most of the people in the population are already getting infected to some extent by the SARS-CoV-2 virus. And in a very recent report, which have come only yesterday in Nature Journal, it has been published that in some African countries, particularly in Kenya, an antibody-based study was done, wherein it was found that 1 in 20 individuals in a country like Kenya have already developed antibodies against SARS-CoV-2. Although in Kenya, number of individuals suffering from the disease was quite low. It was only around probably 28,000. Only around 28,700 individuals suffer from disease, and 460 individuals died so far in Kenya. And in many other African countries also, the mortality and the suffering is very, very low. But on the other hand, 1 in 20 individuals are already showing presence of antibody against the virus. That proves that maybe a large chunk of the individuals, large proportion of the individuals in the country might have suffered from the disease, but they did not develop symptoms. And same may be true in other parts of the globe, including India. And in a report recently published, it also said that maybe by around February 2021, it is assumed that maybe around 12 crore population in India might be showing positive to coronavirus, the COVID-19. So a situation may arise where a large chunk of population in a community or in a particular geographical area may be protected by either exposure to the infection necessarily or by suffering from mild infection without symptom. And at that point of time, necessarily the entire population will attain immunity through herd immunity. Now I come to our humble effort in our fight against COVID-19, and these are two papers recently published by my group of scientists in our effort to develop some strategy against COVID-19. This is one publication which was published very recently last week on 7th of August in Virus Research, a very highly coveted journal. This was on use of codon usage pattern for predicting the adaptability of the virus to different hosts. As the virus has been already isolated from different animals, including dogs, cats, lion, then tiger, etc., in particularly in America, and there has been several reports of infection of animals by COVID-19 virus, with SARS-CoV-2 virus. In this particular study, we wanted to study whether the virus can adapt itself to the dogs and other livestock like cattle, cat, horses, etc. And we found that the virus can adapt itself more to human being than that of other animals, including dog. But out of the animal species, we found that adaptability might be more for dog in comparison to others. That means dogs, pet dogs may also play a role in carrying the infection from infected human being to other susceptible individuals. But of course, this is a theoretical study. This is purely based on bioinformatics analysis. Another analysis based on bioinformatics was also published in Journal of Biomolecular Structure and Dynamics. It is on a particular approach in detecting some biomolecules as a repurposed drug, which can be probably acting as an which can be used for treatment of COVID-19. So this is also a theoretical approach in detecting biomolecules which can be active against this COVID-19 disease. So these are already published, but these are yet to be validated by in vivo experiments. Now, at the end, I'd like to quote a famous statement of Stephen Magee, a brilliant author, who said that smart people stay home when COVID-19 lockdown ends. As we all know, that remaining home or staying at home and keeping social distance and wearing mask, these are the three important tools for fighting COVID-19. So a smart guy or an intelligent person will still remain at home when even the lockdown ends. So we'll be safer remaining home, and if not at all required, we should not go out of home purposelessly so that we can get protected. So this message to you, I'd like to stop my presentation. Thank you very much once again for being with me and listening to the lecture for more than 1 hour. Thank you very much.